Department of Zoology, HPT Arts and Arbiter Science College, B.Sc, Zoology, Semester 1, Mammalian Histology, ZY 334, Paper 2. Presenter's name, Dr. Jawale CS. First, before starting this course, we must get acquainted with the latest university course syllabus of Mammalian Histology. Here, first chapter is Introduction, Definition and Scope of Mammalian Histology. Second chapter deals with the definitions and review of tissue with respect to location, structure and function. Different tissues like epithelial, connective, nerves and muscular. Third chapter deals with the histological study of different organs like skin, where we study in detail vertical section of skin, tooth, where vertical section of tooth will be studied in detail, tongues, sagittal sections with reference to the mucosa, papillae and taste buds. Fourth one is the alimentary canal, where we study basic histological organization of esophagus in transverse sections, stomach in transverse sections, duodenum in transverse sections, ileum in transverse sections, and rectum TS. Then glands associated with digestive system. Here we study histology of salivary gland, parotid, submandibular and sublingual. Histology of liver and pancreas with reference to exocrine and endocrine components. Then respiratory organs, histology of trachea and lung. Then histology of blood vessels like artery, vein and capillary. Next one is the kidney, histological structure of kidney. In that, the detailed structure of nephron and juxtaglomerular complex. Thereafter, histology of reproductive organs that include testes with reference to seminiferous tubules and cells of Leydig. There, in ovary, we'll see histological structure of primary, secondary and matured ovarian follicle, corpus luteum and corpus albicans. And the last chapter deals with the histology of endocrine glands, pituitary gland, thyroid gland and adrenal gland. Overall, it is based on histological detail ultrastructures. The human body is an integrated organism, in the making of which there is a cooperation and integration of parts at different levels of organization. Cells, as vital physical entities, combines to form tissues, tissues to form organs, and organs to form system, and the system ultimately enter into an anatomical arrangement of a human body. These successive grades in a hierarchy of bodily structures and functions comes under the preview of histology. Histology, a Greek word histos, that is tissue, and logia, that is knowledge, is a strict sense the science of microscopically visible components of tissue. In other words, microscopic anatomy or microanatomy. It incorporates three subdivisions like cytology, cells, histology proper, that is tissue, and spatial histology, organology. The organology pertains to the study of arrangement of tissues to form organs. Histology also includes histochemistry, which is the microscopic study of chemical components of the cells and tissues. The term fine structure, submicroscopic structure and ultrastructure used by many workers in the context of histology, related to the detail as reflected through electron microscope. Tissue constitute the matrix of body, the stuff out of which are constructed all those organs and systems which fit together to make up the machinery of a complex living individual. At present, the original concept of tissue based on web or woven tissues of fibers is being interpreted differently. That is, tissues are organized groups of similarly differentiated cells and their derivatives. united in a performance of particular function. Histology, branch of biology concerned with the composition and structure of plant and animal tissue in relation to their specialized functions. The term histology and microscopic anatomy are sometimes used interchangeably, but a fine distinction can be drawn between the two sub-studies. The fundamental aim of histology is to determine how tissues are organized at all structural levels, from cells and intracellular substances to organs. Microscopic anatomy, on the other hand, deals only with tissues as they are arranged in larger entities such as organ and organ system. In their investigation, histologists mainly examine quantities of tissue that have been removed from the living body. These tissues are cut into very thin, almost transparent slices using a special cutting instrument known as microtome. These thin sections, as they are called, may then be stained with various dyes to increase the contrast between their various cellular components so that the latter can be more easily resolved using an optical microscope. Details of tissue organization that are beyond the resolving power of optical microscope can be revealed by the electron microscope. Tissues can also be kept alive after their removal from the body by placing them in a suitable culture medium. This method is useful for culturing and later examining certain types of cells and for studying embryonic organ rudiments as they continue to grow and differentiate. A special branch of histology, histochemistry, involves the chemical identification of various substances in tissue. Basic procedures in histology. Most scientists use the procedure developed by histology during the course of their studies. While histology is an enormous broad field, each researcher typically understands the basic histology of organisms they are working on. The botanist might not understand the human kidney, but plant tissue would be relatively familiar. The practices of histology have extended to nearly every field in science because of how useful they are in preparing and visualizing tissues. Starting with the simple observation using light microscopy, histology has evolved hundreds of different techniques and procedures for staining and observing cells. The most basic form, a simple stain, is achieved by allowing a specialized staining material to wash over cells on a slide. The stains are formulated so that they only stick to certain parts of a cell, such as the DNA. When the stain is washed away with water, only the DNA or other targeted materials remain stained. This allows for better and more advanced viewing of different processes. It was in this way that the process of mitosis was first understood. After technique, sectioning is used in many fields to identify the internal components of cells, often used in conjugations with staining. This technique involves fixing a cell in a solid material so that sections of it can be cut off. At the simplest level, this can be done with an extremely sharp knife and an onion cell, but most applications require more precision. Methods have been invented of replacing the cytoplasm with plastic epoxy, then cutting through the hardened plastic. This method preserves the integrity of an internal components of the cell. Similarly, cells can be frozen and then fractured apart. This also reveals the internal component of the cell without damaging them. These methods, in conjugation with new electron microscopy methods, have led to significant advances in histology and imaging in general. Scientists are now turning to histology to answer fundamental questions in many fields. For instance, the agriculture industry uses the histology of plants to identify early nutrient deprivations, and water usage. The medical industry uses histology to diagnose and cure diseases. Biologists use histology to combat pests and understand the interactions between organisms. Career in histology. Histologists are scientists that specialize in identification of various tissues and cells. Since the invention of microscope, histology has been a field in science. The field has expanded rapidly and the histology of most organisms is understood. Histology is used in a number of professional fields, from lab analysts to medical professionals. Histologists are also used by law enforcement. Histologists are also used by law agencies to help solve crimes. A cytologist is a specialist focusing on the cells found in a bodily fluid, which can often provide DNA evidences and more clues. Histology degrees can be anything from histology certificate program, training you to be a histology technicians to masters and doctorate degrees in histology. Histology technicians usually works in a lab setting, processing samples from hospitals, clinics or research centers. Typically, technicians get to process samples and create slides and do the lab work involved in staining and visualizing structures. Identifying structures and diagnosing disease is taught in higher level program, the highest being postdoctoral training and research in histology. This profession works on the leading edge of science and help create new and useful diagnosis and identification methods. A histologist at this level could start a lab, work for an established lab, or work for a research university teaching and researching. Many medical technology companies, pharmaceutical companies, and other companies required to test their product need trained histologists. Much of the research done on the product to understand their potential for doing harms is done on laboratory organisms. From bacteria to pigs, histologists who specialize in those tissues are needed to understand the complicated changes that some drugs and products may have on the tissues in a body. In understanding what to look for in normal tissue, a histologist can diagnose and theorize about why the product would create a specific reaction in tissue. This feedback is necessary for the biochemist and engineer to tweak their products to ensure they won't be harmful to people. As mentioned earlier, the variety of fields using histology is ever expanding. Histology is a broad and complex field and offers a plethora of opportunities for the motivated student.